you asked me to talk about the biological evidence for biblical chronology, and I think one of the most important biological evidences that we can look at is the idea of the evolution of man. And in fact, I've gotten several questions. Is my microphone on? Okay. I've gotten several questions regarding the evolution of man from people kind of either in question and answer or just randomly catching me between talks. So this will answer some of those, and then I have a little bit at the end, if we have time, to talk about some carbon dating and some radiometric dating that give us a lot of evidence or a lot of reason to believe that the world is a lot younger than the evolutionary biologists say that it is. So I'm going to try to talk a little bit about both those things. And John Wynn's name is on this talk because I am deeply indebted to him for some of these slides and figures, and also the work that he's put together in terms of debunking human evolution. He has a book he's about to publish in a few weeks or a few months on that subject, and he has a tremendous amount of information on it in the appendix of his book, A Catholic Assessment of Evolution Theory. And if you were going to read one book about evolution, that would be the book I would recommend, is A Catholic Assessment of Evolution Theory by John Wynn, because he covers everything from theology to social political things like Mr. Owen was covering in the last talk in terms of the connection with the eugenics movement. And he has two giant appendices, one on human evolution and one on all the icons of evolution I presented in my first talk. This is an excellent, excellent resource, and I owe him a deep debt of gratitude here. So without further ado, we're going to look at paleontology and molecular biology and ask the question whether it's more reasonable to believe that there was a historical Adam and Eve, or more reasonable to believe that millions of years passed and we descended from primates. So I'll start with a quote by Ken Miller, who is someone whose name makes me twitch a little bit. Claims to be Catholic, but supports an absolutely atheistic idea of evolution. And he says, for all the fuss and concern that surrounds the idea of having human evolution, the detailed fossil evidence of our ancestry is remarkably powerful. Well, we're going to take a look at this detailed fossil evidence. And of course, I had to throw on there with a TV Man instead of McDonald Man. But John's put together a timeline here for us that shows some of the more commonly cited human ancestors. There's a few that are not on this table, but we're going to look mostly at Homo erectus, which is right below Homo sapiens, but Lucy's on there, Australopithecus afarensis, and the Tong child is on there, Australopithecus, which I can't say, obviously, africanus. And there's a couple of other things that are on there in terms of human ancestors, and we won't deal with them specifically, but if you're interested in the specifics, I do refer you to those two books by Mr. Wynn. So we're going to look at Homo erectus and see if the fossil evidence gives us a tremendous amount of reason to believe that man evolved from apes or ape-like ancestors. So Homo erectus was discovered in 1891 by Eugene Dubois, and he found a very primitive skull cap and a modern-looking femur, and put the two of them together and said, oh look, I found the ancestor of man. So somebody who would have been more primitive than man, but more evolved than the apes. And it turns out that these two fossils came from two different beds that were several dozen feet apart from each other. So why he thought the two came from one organism is still a little bit of a mystery to science, but that's okay. He identified what he thought was a new species, and there have been a number of other fossil finds that have sort of corresponded with what he had. So he noticed that the cranial capacity was lower than that it's normally seen in humans, and he emphasized the more primitive morphology of Homo erectus, which is sometimes exaggerated. So these are 4 Homo erectus images that I found on Google a couple of days ago, so they're pretty recent. The one furthest to the left is from the Smithsonian. The two in the middle are from websites. One is devoted to promoting scientific literacy, and the other one is devoted to promoting evolution specifically. And then the final one on the far right is from a Spanish website, so I don't know what they were promoting, but that was Homo erectus. So you can see that in some cases the drawing looks almost human, and in other cases it looks very ape-like, and certain artist renderings emphasize the primitive morphology of Homo erectus to sort of connect us with the apes. And as we saw with our whale intermediates, there's a little bit of artistic license going on here. So was Homo erectus like us, or like apes, or somewhere in between? Well, if you look at specific bones, so for example the clavicle, which is located in your shoulder, the clavicle of Homo erectus fossils fall within the normal range of human variation. And in fact, it looks like Homo erectus could have engaged in high-speed throwing. So they might have played baseball, which doesn't sound very chimp-like. And they had an essentially modern human foot. And they walked upright just like we would walk. So they could have run around the bases as well as throwing and hitting the ball. Their body size was essentially modern. And one of the famous fossils that was found, Narikatome boy, would have grown to between 5'9" and 5'11" in height and weighed between 176 and 183 pounds if he had grown up to maturity based on the estimates of the archaeologists. And so we're seeing about normal body size for Homo erectus too. So indistinguishable from man in the clavicle, indistinguishable from man in the feet, indistinguishable from man in body size. And if we're looking at body size in that evolution of man icon I put up earlier, just like with the evolution of the whale, you see this nice progression from the little bitty monkey thing to the nice big man, and of course the man in front of the TV. But what you actually see if you plot out the sizes of all of these supposed ancestors of man is you see them getting smaller and staying the same size, and then getting bigger, and then getting smaller, and then getting bigger, and then getting smaller again, which doesn't really look like evolutionary progress to me, right? Now, to a certain extent, it's not fair to suggest that there's such a thing as evolutionary progress, because any evolutionist worth their salt will tell you, well, we're not progressing towards something. But they will also tell you that this whole kind of U-turn up and down thing, that is a little odd. And it looks a lot more like that idea of oscillation around a mean that we were talking about earlier than it looks like any kind of forward evolution from one species to another. Okay, and also about human body size. I like to point out to people, if we're just going on body size, if you buried Shaquille O'Neal and Danny DeVito next to one another and somebody dug them up in 1,000 years, they would probably think they were different species. But they would be wrong, because they both fall within the normal variation of human beings. And what we're seeing with these bones of Homo erectus is that they're falling, at least in terms of size, within the normal range of normal human size. Right? They're also falling in the range of normal human size in terms of their cranial capacity. Now, their average cranial capacity is lower than humans' average cranial capacity, but it's still well within the range, which is about 900 to 1,200 cc's or milliliters. And if you look at group means, the modern human capacity ranges from about 1,000 to about 1,500 cc's, and that fits pretty well actually with the Homo erectus. They just fall on they fall into a lower group in terms of modern human cranial capacities. So I was thrilled to death when I read this slide the first time. This was published in Nature, and if you're familiar at all with biology, you know that the journals Science and Nature are the Bibles of biology. This is where all the best research gets published, and if it gets published in Nature, it is the thing, period. So I was thrilled when I read this, because I remember traveling to, and I'm actually going to go back to the previous slide. I remember traveling to a natural history museum, and they happened to have a human evolution exhibit, and this was while I was still kind of in my, I'm not sure about wonky, but I guess it's, what everybody accepts, so okay. And I noticed that a lot of the skulls looked like that one in the picture, where there was a lot of brown and then a little bit of white. And so I started reading captions, and it turns out that what was white is what was actually dug up, and what was brown is what was filled in. Okay, and I was walking around this human evolution exhibit going, this stuff is 80% brown. They made it up. And from that point on, I really didn't have any problem with thinking that there really weren't any good human ancestors in this supposed link. And then when I started discovering things like this, I felt very vindicated, because I thought to myself, well, you're seeing these skulls and they look sort of human, but they look sort of not, but they don't look really inhuman or unhuman. And maybe there's just something wrong with this particular individual that's causing their skull to be shaped this way. So in Nature, this team published that there were 7 different explanations why Homo erectus morphology resembles Homo sapiens morphology. And they said that the Homo erectus could have been due to inbred communities. It could have been due to nutritional problems, low-grade anemia, genetic factors, hormonal factors, some kind of pathological condition that caused their skull to be misshapen, or just natural variation in bone thickness that provides a better chance of being preserved, leading to the false conclusion that the whole population was thick-boned and of a different species. So there are normal environmental factors and genetic factors that can explain why you're seeing populations of individuals that are within the normal range of humans, but they tend to cluster around a slightly different average, a slightly different mean. And so if we look at cranial capacity, the same way we looked at body mass, you'll see that there's that U-turn again. So the brains are getting bigger and then smaller and then bigger and then smaller. And that Homo sapiens brain is actually smaller than a Neanderthal's. And cranial capacity is touted a lot in human evolution and as a measure of intelligence. So the higher the cranial capacity, the more intelligent something is supposed to be. So you're dumber than a Neanderthal. And if you buy this argument, men are smarter than women because they have larger cranial capacities. Now, most evolutionary biologists don't go that far because that's very politically incorrect. But if you're going to tout cranial capacity as a measure of intelligence, then you're going to have to acknowledge that men are more intelligent than women. It's a very unpopular point of view. And also that you're dumber than a Neanderthal. So cranial capacity, it may or may not be an indicator of intelligence. Probably actually is not necessarily an indicator of intelligence. And the things that have higher cranial capacity, you'll note that Homo erectus, again, falls within the normal range of man. And then most of the stuff down here falls within ranges that are pretty comparable to each other. So it's possible that we're drawing these lines and connections between things that are actually not different species. And lending even more credence to the idea that Homo erectus and Homo sapiens are not a different species is the fact that Homo erectus dates to only about 10,000 years ago, at least according to evolutionary biologists using their fossil record scheme. So Homo sapiens are supposed to have evolved about 2,000 years ago. So that means that we coexisted for about 1,900 years. Now, if something is the ancestor of something else, they don't coexist for almost 200,000 years in the same place at the same time, occupying the same ecological niche. That's not how evolution is supposed to work. So this results in what's called a contemporary status problem. So these two things lived at the same time, so they probably, one can't be the ancestor of the other. And this also lends credence to the idea that Homo erectus are simply human beings who have some kind of genetic or nutritional problem. I lost my water. Okay, now just to see if everybody's paying attention, can you identify the Homo erectus skull? Ooh, who said that? You are so right. All of them. That were unearthed in Georgia between 2000 and 2013. Now, I'm not an archaeologist or a paleontologist, and so when I first looked at that, I was like, oh, those look like they could be different species to me. So do you see where some of the confusion is coming from? Right? We're digging up skulls that look human, but there's a wide variation in human skulls, and there's a wide variation in so-called Homo erectus skulls. And it's actually kind of hard to tell if something is a different species from something else based simply on its skull. Teeth are actually even worse because a number of characteristics of teeth are controlled by a single gene, such as cusp number and even the number of teeth. So if you're classifying things based on teeth, that's a particularly suspect way to classify them, because a single gene mutation can result in very different teeth, and a single gene mutation does not always result in a different species. So the Georgia team concluded basically that there's so much variation in the fossil hominids that tends to be interpreted as species diversity and not diversity, so variation within the species. And they're actually calling for a single evolving lineage from about 1.8 million years ago to about now that they would call Homo erectus. And they're lumping a couple of other things into there too. Homo habilis and Homo rudolfensis, they say, belong to this single Homo lineage. So there are a number of evolutionary biologists, because of these morphological characteristics that fall within the normal range of human variation, who are calling for what's called the sinking of Homo erectus, which means that they think that Homo erectus is not a separate species, it should be collapsed into Homo sapiens. And this is partly due to the fact of that contemporary status problem. You have Homo sapiens and Homo erectus fossils that occur in the same community, so living side by side, or at least buried side by side, at the same location for extended periods of time. You have fossils that display features of both Homo sapiens and Homo erectus. Scientific literature admits that there are many non-evolutionary factors that can account for the variation you see in Homo erectus, and even notable evolutionists basically have said Homo erectus is really not a good classification. Really, it's all Homo sapiens. It falls within the normal range of variation. They're just people, right? So there are more sinkings that have been called for, and I'm not going to go through each of them individually, but Homo ergaster, Homo antecessor, and Homo heidelbergensis, they're all very similar to modern man and share certain features in common and fall within the range of normal human variation. And so it's very likely that these are not different species. They are the same species. And we can't really test that from a biological species definition because you cannot determine whether fossils can reproduce for obvious reasons. And then, of course, the last one is Neanderthal Man, which I think is quite interesting. Neanderthal Man, and I don't know how they got Neanderthal Man DNA. That's probably something I need to look up. But they sequenced some Neanderthal Man DNA, and they discovered that Neanderthal Man experienced some gene flow back and forth, and that would indicate that they were able to successfully reproduce, and if we are able to successfully reproduce, we're the same species. So Neanderthal Man should definitely be sunk. And if you do that, you do all those sinkings, you end up with a timeline that looks like this. It's been a Homo sapiens from the beginning. Homo sapiens, and nothing but Homo sapiens. And some of the Australopithecines actually should be sunk into orangutan, not into Homo sapiens. So it's a little more complicated than the very brief overview I've just given you, but again, I refer you to John Wynn's book for the rest of those details. So there's genetic evidence, right? And you've probably seen claims that you're 98% chimp.
And 50% banana.
And 50% banana, yes, you learned that earlier today. So let's look at that. So initial estimates of human-chimp dissimilarity were given between 3.9 and 13%, and this was published in the journal Nature and then the Proceedings of the National Academy of Sciences. So large. And even if it was only 3.9% difference, the differences between your genome and a chimp's genome would be about 117 million base pairs. Now, it's too big of a number for us to really conceptualize, so that's about 29,250 pages, which is more than Dr. Syngenis has written in his entire lifetime. And unfortunately, he's not here for me to take that jab at him personally, but I have in the past, so no gossip. Given evolutionary estimates of the divergence between humans and chimps, which was approximately 300,000 generations ago, that means that we need about 390 mutations per generation. The current rate of mutation in humans and chimps is about 100 mutations per generation. So even if you count both species, you only get about 200 mutations per generation, which is only about half of what you need to see the divergence that you have if humans and chimps diverged as long ago as they were supposed to. And that's if you only have a 3.9% difference. Have you followed all my math so far?
Yes.
Okay, good. I'm used to teaching students who don't so much follow my math, and I talk too fast. Okay. So if we have a 13% difference, we have now quadrupled that problem. Roughly, slightly more than quadrupled that problem. Okay. If we look at specific subsets of the DNA, so for example, microRNAs are tiny bits of RNA that are produced, that are part of that all-important regulatory process I was describing earlier that determines when genes get turned on and turned off. And they looked at microRNA expression in the brain and found that of the 447 microRNAs that they discovered, chimps had 25 that humans did not, and humans had 51 that chimps did not. And so even ignoring any kind of sequence difference in the ones that we had in common, that's an overall difference of 17%, just right there in our microRNAs. So even if we're not seeing a difference so much in the whole genome, we're seeing a tremendous difference in the way that these genes are expressed, and what kinds of genes are expressed, and what kind of regulation is going on here, which is why you can write a sonnet and a chimp cannot. So when you're comparing human and chimp genome similarities, there's a really important thing you need to know, especially about those early estimates. And it involves the very complicated process of shotgun sequencing. So this is what I was doing at Washington University in St. Louis ever so long ago when I had my first biology job. And you would take the whole genome of an organism, which you can kind of imagine as a colored line, that colored line at the top. That's all in a row and all ordered correctly. And then as a scientist, you chop it up into little pieces, approximately 2,000 base pairs. That was my job. A fun machine that had like a ruby in it that cut the DNA down into those shreds. And then you had to kind of process it all, get it into bacteria, send it off to be sequenced. And once the sequence is obtained, the people who had the really hard job were called the finishers. And they took all of those 2,000 base pair sequences and they had to line them up. And they said, well, there's an overlap of about 200 base pairs here, so this fragment must go with this fragment. And that's where you see kind of, you see them all mixed up in the middle. You see them kind of starting to get aligned there at the bottom. And you see the final sequence that we come up with at the very bottom. So this is very tedious. You can imagine it's kind of like building a 1,000-piece puzzle, except 10,000 times that hard, because you don't know which are edge pieces, right? And there's a lot of repetitiveness in the DNA too. So this took about a decade to finish completely. And then it also turns out that just a few months ago, or maybe about a year ago, I think it was last November, they published some data on the fact that, we thought the human genome was 3 billion base pairs, but actually 3.5 billion base pairs. We lost 500 million base pairs in there trying to do this whole process. That was a little embarrassing. So when researchers went to sequence the chimp genome, they said, well, that's tedious, time-consuming, we don't have the time for that. We know that humans and chimps are related, so we'll just line the chimp genome up against the human as a template. Do you see the problem with that? So when we see similarities in human and chimp genomes that are quoted before the last couple of years, they're comparing this chimp sequence that was actually formed by lining it up with the human sequence to see what order the pieces were supposed to go in. So that's called an artifact of your method. That was bad science. And it was such bad science that even the evolutionary biologists admitted this chimp sequence is terrible. We need to resequence it. So they've resequenced it. They've published, I think, about 80% of it at this point, or at least at the point of the last paper that I read, which may be a year or two old. And based on that's been published so far, Dr. Bergman and Dr. Tompkins have done some blast analysis, and they have concluded that actually we're at least 17% different from chimps. 17% is a lot more than 2%, especially if to get to 4% we needed twice as many mutations as were actually occurring in chimps and humans at this moment. At this point, we've compounded our problem somewhat ridiculously. All right. As if that wasn't bad enough. There's more. So there's some tremendous dissimilarities in the Y chromosome. The chimp Y chromosome was one of the first ones that was sequenced. It's very small. It's half the size of the human Y chromosome, which is already pretty small. And the half that was sequenced is only about 70% similar to the human Y chromosome. So that's an overall total similarity of 35% between the Y chromosome of humans and the Y chromosome of chimps. And researchers have admitted that they think that this is the kind of dissimilarity they would see between birds and humans, not between their supposed closest ancestor and humans. So this is problematic. This is even more problematic when you look at the Y chromosome just of man. So of course you get your Y chromosome from your dad, and only men have Y chromosomes. And scientists have actually traced the Y chromosome lineages back to a single Y chromosome, and they've named him Y chromosome Adam, and then they immediately regretted naming him that. Because they wanted to say he lived a really, really long time ago, and he did not live at the same time as Mitochondrial Eve, who we'll meet in the next slide. But it turns out that they were contemporaries. So that's problematic. But the Y chromosome of most modern men is less than 500 mutations removed from Y chromosome Adam. So that's not very many mutations, and this amounts to about a 0.002% change, which is not very much of a change. And if we have one mutation in the Y chromosome per generation, and remember we've got 100 mutations in the whole genome per generation, and we've only got 23 pairs of chromosomes. So one in the Y chromosome is really not that unreasonable. That means we've got less than 500 generations, max of 10,000 years, to produce all the Y chromosome variation that we observe in modern man.
Uh-oh.
Yeah.
This is in trouble.
It is in trouble. And this is absolutely incompatible with the high mutation rates that would be necessary to produce the divergence we see between chimps and humans. So we can't have it both ways, ladies and gentlemen. We can't have humans being so similar in their Y chromosome that it would only take 10,000 generations to produce that difference, and humans and chimps being so different that it would take as long ago as birds are supposed to have diverged from mammals. The mitochondrial Eve compounds the problem. So if there was an Adam, was there also an Eve? And yes, there was. Dr. R.W. Carter analyzed 800 individuals' mitochondria and identified a probable consensus sequence for an original mitochondrial DNA molecule. And prior to Dr. Carter's analysis, which was fairly recent, they had identified a number of mitochondrial lineages, and you could sort of send a DNA sample off to one of these 23andMe type places and figure out which mitochondrial DNA lineage you were following. And it actually makes a little bit of sense if you think about the flood lineage and possibly several mitochondria. It was Noah and his wife and his sons and their wives on board the ark, and the sons were all related, but the wives were not. So it is possible that we have slightly more mitochondrial variation there because we have a bottleneck. So the average human being only diverges from this consensus sequence that Dr. Carter identified by about 22 base pairs, but up to about a max of 100 base pairs. And mitochondrial DNA mutates slower than regular DNA because there's less of it and it's more condensed, and mutations tend to be more fatal, so you don't get to pass them on. But it mutates at a rate of about half a mutation per generation, and this would mean that at the most we would need 200 generations, which would also be less than 10,000 years, to achieve the mitochondrial DNA diversity that we see today in modern man. So both Y chromosomes and our mitochondria are telling us that really human beings are much too similar to have evolved millions of years ago. So in conclusion, despite lofty claims, neither the paleontological evidence nor the genetic evidence support the idea that there was a transition from an ape-like ancestor to modern man, and it's actually pretty consonant with man being unique in creation and having been recently created. There's also evidence that all the variation we see in man could have come from a single historical couple less than 10,000 years ago, which I think is pretty exciting. So once again, evolution fails. And I won't go through my list because this was a fairly short talk, so hopefully you remember it all. But I found it really remarkable that this I think he's Homo Neanderthalensis. He's either Homo Erectus or Homo Neanderthalensis. He looks remarkably like Patrick Stewart. Remarkably like Patrick Stewart. Okay, and so for once I'm not over time, so I do actually get to talk about the age of the Earth, which is very exciting. Okay, we're going to do a brief review of chemistry and radioactivity. We're going to talk a little bit about radioactive dating methods, including carbon dating and other radio dating, and then we can come to a conclusion about whether the Earth is really as old as it looks. And if someone asks you or tells you that the Earth just looks old, it's a good idea to ask them, well, why? Why does the Earth look old? What would an old Earth look like? I don't know. All right. So atoms and isotopes. Most atoms have a variety of isotopes, and this means that they have masses, and they have different masses because they have different numbers of neutrons in their nuclei. And this matters because some configurations with certain numbers of neutrons are stable, and other configurations with different numbers of neutrons are not. So carbon-12 and carbon-13 are stable. You will find those in the environment. They will stay carbon-12 and carbon-13 until the end of the world. But carbon-14 is not stable, and it will not stay carbon-14. It's radioactive, and it will decay into a different kind of atom. So it's produced by electromagnetic radiation in the atmosphere. So electromagnetic radiation will hit a nitrogen-14, and it will cause the conversion of one of those protons into a neutron. And then you get nitrogen-14 being transformed into carbon-14, and then carbon-14 is radioactive. And carbon-14 does not stay carbon-14, as I just said. It will spontaneously convert back to nitrogen-14. And so we have what is known as the parent element. This is the carbon-14. And then the daughter element, which is the nitrogen-14 that's formed when the parent element decays. And this decays in a very strange way. So we'd say that radioactive elements have half-lives. So the half-life of carbon-14 is about 6,000 years, and that means that about half the sample will have decayed back to nitrogen in about 6,000 years. So suppose that I have about 100 atoms of radioactive carbon-14. How many atoms would be left after one half-life? Let's see who's paying attention. Go ahead, sir. No, 0 is wrong.
50.
Oh, 50. Yes, sorry. Misinterpreting your animations. All right, how much would be left after 2 half-lives? 25. Good. This is where my chemistry students always mess up. They always go, none. And I'm like, no. Half the sample. So if you start with 50, after 6,000 more years, you'll have 25 left. So you end up with a radioactive decay curve that looks something like this. Right? And this can tell you something about how old an organism is if you know how much carbon-14 it had in it to begin with, and you know how much carbon-14 it has in it now. Because organisms consume carbon-14. The carbon-14 that's produced in the atmosphere is taken up by plants. They make it into radioactive glucose. So then the plants themselves are radioactive, and then cows consume the radioactive plants, or other animals consume the radioactive plants, and you consume a hamburger, and now you're radioactive too. So this is how carbon-14 gets into everything, from the atmosphere to the plants, from the plants to the animals. And while you're alive, you're continuously taking in carbon-14, and you're also giving off carbon-14, and it's also spontaneously decaying, but it stays at a relatively constant rate inside your body because you're taking it in and giving it off. When you die, you don't eat anymore. I know this is a shock. But you are not taking in any more carbon-14, so the only thing that's happening is the carbon-14 is leaving your body by radioactive decay. So if we know what percentage of carbon-14 is in the atmosphere at the time that you die, we should know what percentage of carbon-14 was in your body at the time that you died, and we can calculate how much carbon-14 is in your body now, and compare the difference between those two, and see how many half-lives has passed. Is generally, I just said my whole next slide, because you know, whatever. All right, and I mentioned before there's a theoretical limit to this. The theoretical limit is about 100,000 years. The actual limit, according to Beta Analytics, which is a pretty good website, they set their limit at 43,000 years, but it's somewhere in a range between 42 and 53,000 years. They like to be conservative, which makes sense. So now we come to. So in a lot of cases, carbon-14 isn't present when it should be. So living things should have carbon-14 in them, but living trees growing next to a German airport dated at us having died 10,000 years ago because they had that little carbon-14 in them. Plants growing in Montezuma well water that was depleted of modern carbon-14 dated to about between 17,000 and 24,000 years dead. And a live plant subjected to gas from an ancient carbon source dated to about 4,800 years dead. So sometimes organisms can be living in environments that are low in carbon-14, so then when you do a carbon dating on them, they actually date to older than they are. And this is one of the reasons why it's possible to reconcile those carbon dates I had on the dinosaurs earlier of 28 to 32,000 years with a 10,000-year-old Earth, because they could be dating older than they actually are. Now, this is fairly common in marine mammals. Freshly killed seals were dated at 1,300 years dead, and some living mollusks were dated at 2,300 years dead. And talkorigins.org, which is one of the primary pro-evolutionary websites and blogs out there, they have a lot of really good information. I usually go to them to see what arguments are being made against creation arguments, because I don't want to use an argument that's invalid. And they point out that these aquatic animals are feeding off of other animals that are living in a nutrient-rich upwelling zone, so water's coming up from way deep in the ocean, and this water that's way deep in the ocean is depleted. That these marine animals would date to older than they are, and that's really not a problem for evolution. But marine animals aren't the only thing that date older than they are, and so I don't think this mortar in this castle was eating things from the upwelling regions, and it's still dated at 6,000 years older than it actually is. So there are a number of these different dates you can find where carbon dating has gone wrong. And why would that happen? Well, we're making an assumption that at the time this organism died, or at the time this castle was built, that the carbon-14 percentage in the atmosphere is the same as the carbon-14 percentage in the atmosphere now. And we actually know that assumption is untrue, because carbon-14 has not yet reached equilibrium in the atmosphere, so its concentration is still changing. If its concentration is still changing now, or 2,000 years ago, or 6,000 years ago, and it's highly unlikely that the modern percentage of carbon-14 is going to be identical to the historical percentage of carbon-14. So if we don't know the percentage of carbon-14 we started with, it's actually impossible to carbon date anything, which is kind of sad. Okay, and Dr. John Sanford, writing a book, Contested Bones, with his co-author, pointed out that it's remarkable, but essentially everything in the fossil record contains measurable levels of carbon-14. Now, unless you're a biologist, this probably isn't as earth-shattering to you as it is to me. But basically what that means is that everything in the fossil record is less than 43,000 years old. Coal, dinosaur bones, everything that's supposed to be millions and millions and millions of years old is less than 43,000 years old. And he says this seems contrary to popular wisdom, but is widely recognized within the carbon-14 community. Taken at face value, this suggests the entire fossil record is less than 100,000 years old, 43,000 really if you're going with the practical limit rather than the theoretical limit. Many will be offended by this idea, but like it or not, this is what the carbon-14 data appears to be showing. So something here appears to not be as it should be. There are other radiometric dating methods, and we have these other radiometric dating methods because not all rocks have carbon in them. So we're looking at other dating, so potassium, argon, uranium, lead, and rubidium, strontium. They work by similar methods. You have a parent element that decays into a daughter element, and if you know the starting concentration of your parent element and you know the final concentration of your daughter element, you should know how old this thing is. But it's based on three key assumptions, and Mr. Owen mentioned these earlier. You have to assume that there was no daughter element in the rock at the time that it formed. You have to also assume that the rock is a closed system, which means that no parent into the rock and no parent or daughter element could leave the rock. Now, any rock that's exposed to water at any point is not a closed system, because things dissolve in water and they can be introduced into the rock or removed from the rock. And the third assumption is that the rate of decay has been constant for the rock's entire history. And there's some good reasons to believe that that might not be true, but they're a little bit above my pay grade, so I don't like to present on things that I don't entirely understand. So I would refer you to a book, Thousands Not Billions. I believe it's by Dom DeYoung. And he talks about some of the things going on here with the rate of decay. So that's three big assumptions. And you know what happens when you assume. You become an evolutionist. All right, so the reliability of these other types of radiometric dating is actually significantly less than the carbon dating. The carbon dating is more reliable because we can actually calibrate it, at least to some extent, with things of known age. Can't really do that with the other kinds of radiometric dating because their half-lives are so much longer. So Dr. Morris has noted that when the same rock is dated by more than one method, it will often yield different ages. And when the rock is dated by more than one method, or more than one time by the same method, it also often gives different results. And this leads to problems. So rocks from a 1984 eruption of Mount St. Helens dated to 2.8 million years old when they were less than 20. Radiometric age of a lava flow in New Zealand from 1949, 54, and 75 dated to 3 million years old. This one's hard for me to read. And an eruption in Hawaii in the 1800s dated to being 1.4 to 22 million years old, depending on the dating method that was used and which part of the rock was dated. And there's an explanation for why lava dates so strangely, because magma melts some of the surrounding rock. And so older rock can be incorporated into the magma, and it can be deficient in parent element and rich in daughter element, and this can lead to weird dating ages. Oh, that is Don DeYoung, who wrote Thousands Not Billions. Very good. I'm glad my slides can correct me when I'm possibly wrong. And then you end up with additional problems like upside down dates. So for example, in the Grand Canyon, there's a layer that dated, a lower layer that dated younger than a higher layer, which is a problem for evolutionary biologists who claim that the lower layers were laid down first. So somehow the lower layer was laid down about 50 million years after the upper layer. I don't know who dug it up to do that, but that's what the dating says. And we're at our last slide on radiometric dating, which is forms from beta analytics, the University of Waikato, if I'm saying that right, radiocarbon dating laboratory, and the Kiev radiocarbon dating laboratories. And I've highlighted some important things for you on these forms. So beta analytics asks for the stratigraphic and environmental details, so they want to know what rock layer you found it in. Right, and the University of Waikato asks for the stratigraphic data, but also asks for your estimated age range. They want to know how old you think the rock is before they tell you how old they think the rock is. Right, and the Kiev laboratory asks the same thing. What period is that? Is the Bronze Age or the Neolithic or this, that, or the other? So we have a methodology problem here. They're not objectively dating the rocks, because remember, if you date the rocks using different methods, you get different dates. If you date it using the same method at different times, you get different dates. So what they do is they say, well, where was it found? How old do you think it is? So we'll give you the date that best corresponds with your guess, basically. So this becomes kind of a self-fulfilling prophecy. You think it's supposed to be 2 million years old, and the date that's 3.3 million years old, we'll give you the 2.2 million years old. So radiometric dating basically fails to yield accurate results for objects of known ages. It fails to completely account for the three major assumptions that can completely invalidate the dates, and it's dominated by methodology problems that basically make the process self-affirming. So now even MacGyver is face-palming. And that's my last joke for the day. And I'm on time. Look at me go.